Right now, inside every single cell of your body, there are millions of tiny structures working nonstop, cutting, copying, building, and recycling, without you ever telling them to. You are not conscious of any of it. Your heart beats, your muscles move, your immune system fights, and your brain thinks, all because of machines so small they make a grain of sand look enormous. Here is the number that tends to stop people. You have roughly 30 trillion cells in your body. And inside almost every one of them, there is an entire city's worth of specialized structures, each one doing a precise job all at the same time. These structures are called organelles, meaning little organs, and once you understand what they do, you will never look at a living thing the same way again. In this video, we are going to tour the inside of a cell. We are going to meet the key players: the nucleus, the mitochondria, the ribosome, the endoplasmic reticulum, the Golgi apparatus, and the lysosome. Each of these organelles has a role so specific that losing just one type would shut the whole system down. By the end of this, you will understand not just what each organelle does, but why it has to exist at all, and how they work together as one seamless living machine. We are going to keep it simple, clear, and genuinely interesting. Let us get inside. The nucleus is not just the biggest organelle in most cells, it is the one in charge of everything. Think of the nucleus as the cell's headquarters. It is the room where all the blueprints are stored. Inside it, you will find DNA, deoxyribonucleic acid, which is the cell's complete instruction manual. That DNA contains the code for building every protein your body will ever need. Proteins are the workhorses of life. They form your muscles, carry your oxygen, fight infections, and speed up chemical reactions. The nucleus does not just store that code, though. It reads it and sends out copies. When the cell needs a specific protein, the nucleus makes a molecular messenger called mRNA, messenger RNA, and ships it out into the rest of the cell. That messenger carries the instructions from the blueprint to the builders. The nucleus is surrounded by a double membrane called the nuclear envelope, and it is dotted with thousands of tiny gates called nuclear pores. These pores are not just holes. They are highly selective doorways that control exactly what goes in and what comes out. Think of the nucleus like a protected government archive. The original documents, the DNA, never leave the building. But copies of specific pages, mRNA, are sent out to factories across the city. The pores are like the security checkpoint at the door, deciding what is allowed through. There is also a smaller structure inside the nucleus called the nucleolus. This is where the cell manufactures ribosomes, and we will meet those in a moment. One remarkable thing: mature red blood cells have no nucleus at all. They eject it before entering the bloodstream, which frees up space to carry more oxygen. It is a trade-off. The cell gives up the ability to repair itself in exchange for extra room to do its single job. You have almost certainly heard that the mitochondria is the powerhouse of the cell. There is a part of that story most people never get to hear, and it changes everything. Mitochondria are where the cell converts food into usable energy. The energy currency of the cell is a molecule called ATP, adenosine triphosphate. Every time your muscle contracts, every time your nerve fires, every time your body does anything at all, it spends ATP. And your mitochondria are constantly producing it. They do this through a process called cellular respiration, essentially controlled combustion. Glucose and oxygen go in and ATP comes out, along with carbon dioxide and water. That is why you breathe in oxygen and breathe out carbon dioxide. The trade is happening at the mitochondrial level, in every cell, every second. Picture the mitochondria as a hydroelectric dam. Electrons from broken-down food molecules flow through protein channels in the inner membrane, like water-turning turbines. That flow of electrons powers a remarkable molecular motor called ATP synthase, which literally spins to snap together ADP and a phosphate group, producing ATP. Energy in, energy stored. Ready to spend? Now here is the part that will genuinely surprise you. Mitochondria have their own DNA, not the same DNA as the nucleus, their own separate circular DNA. It looks more like bacterial DNA than human DNA, and they reproduce on their own, independently of the cell dividing. Why? Because mitochondria were not always part of our cells. About 1.5 billion years ago, an ancient cell engulfed a free-living bacterium, probably an aerobic, oxygen-using microbe. Instead of digesting it, the cell kept it. The bacterium provided energy. The cell provided protection. Over billions of years, they became inseparable. That bacterium became the mitochondrion. This idea is called the endosymbiotic theory, and it was formally argued by biologist Lynn Margulis in a landmark 1967 paper. First, the scientific community largely rejected it. Today, it is one of the most solidly supported ideas in all of biology. Your own mitochondria are essentially ancient bacteria that decided to move in and never leave. If the nucleus sends the blueprints and the mitochondria supply the energy, then the ribosome is where the actual building happens. Ribosomes are the protein-making machines of the cell. They receive the mRNA message that came from the nucleus, read it 3 letters at a time, and use that code to assemble a chain of amino acids, building proteins one piece at a time. Here is a number that puts their importance in perspective. A single human cell can contain up to 10 million ribosomes. 10 million. And this machinery is so energy-hungry that producing and running ribosomes accounts for more than 60% of a cell's total energy budget. That is how central protein production is to everything the cell does. A ribosome is tiny, just 20 to 30 nanometers across. You could fit about 1,000 of them across the width of a single human hair. Their size is deceptive. Each ribosome is a 2-part molecular machine made of RNA and protein, and it is extraordinarily precise. A single wrong amino acid can turn a useful protein into a useless or even dangerous one. Think of an mRNA strand as a scrolling printout from the nucleus, and the ribosome as the printer that reads it. As the mRNA moves through, the ribosome reads the code and adds the corresponding amino acid, like pressing the right key for each character. Dozens of ribosomes can read the same mRNA simultaneously, each at a different position, producing multiple copies of the same protein at the same time. Now some ribosomes float freely in the cell's fluid interior, the cytoplasm, building proteins that will be used inside the cell. Others attach to a membrane system called the endoplasmic reticulum, or ER. The ER is a vast folded network of membranes that extends throughout the cell. Where ribosomes are attached to it, it is called the rough endoplasmic reticulum, rough because it looks studded and bumpy under a microscope. These ribosomes make proteins destined for export, for the cell membrane, or for delivery elsewhere. The smooth endoplasmic reticulum, which has no ribosomes, handles a different job. It synthesizes fats and lipids, and in cells like liver cells, it helps detoxify harmful substances. Think of the ER as both a manufacturing floor and a transport highway. Everything that gets made here moves somewhere specific next. Proteins do not just leave the ER and wander around hoping to find the right place. There is a dedicated sorting station and a dedicated cleanup system. The Golgi apparatus was discovered by Italian scientist Camillo Golgi in 1898. It looks like a stack of flattened membranes. Imagine a pile of deflated balloons. Its job is to receive proteins and lipids from the endoplasmic reticulum, modify them, often by adding sugar molecules to them, and then package them into small bubbles called vesicles. Those vesicles are labeled and shipped to their destinations, the cell membrane, a lysosome, or outside the cell entirely. The Golgi apparatus is the cell's post office. Packages arrive from the ER. Workers inside stamp them, modify the address label, sort them, and load them onto the right delivery truck, which is the vesicle. Each truck knows exactly where to go. Now, some of those deliveries go to the lysosome. The lysosome is one of the most underappreciated organelles in biology. The lysosome is essentially the cell's recycling and waste disposal center. It is a membrane-bound sac filled with powerful digestive enzymes, about 50 different types. These enzymes break down worn-out organelles, damaged proteins, and even bacteria that the cell has engulfed. The lysosome maintains a highly acidic interior, much more acidic than the rest of the cell, to keep those enzymes active. The membrane of the lysosome is critical. Without it, those same enzymes would destroy the rest of the cell. The membrane acts as a firewall, containing some of the most chemically aggressive tools the cell possesses. Christian de Duve discovered lysosomes in the 1950s, and he received the Nobel Prize in Physiology or Medicine for this work in 1974. At the time, the idea that a cell contained a built-in self-destruct compartment seemed almost dangerous, but it turns out that controlled destruction and recycling are just as vital to life as construction. So here is the picture. In every cell of your body, and you have about 30 trillion of them, there is a nucleus directing operations, sending out instructions. There are mitochondria generating the energy to carry those instructions out. There are millions of ribosomes reading those instructions and building the proteins that make everything work. The endoplasmic reticulum processes and ships those proteins. The Golgi apparatus sorts and stamps them for delivery. And the lysosome makes sure nothing broken gets left to cause damage. These are not metaphors. This is literally what is happening in your body right now as you watch this. No conscious effort required. Billions of years of evolution have built a system so precise and so cooperative that we are still working to understand the full details. Cell biology is not just about memorizing names for an exam. It is about understanding the actual machinery of life. And once you see it this way, not as a list of organelles, but as a coordinated city that never sleeps, it becomes one of the most remarkable things you can possibly know. If this gave you a new way of looking at how life works, share it with someone who needs to hear it, and we will see you in the next one.